Hi, this is Miss Litton, and this is my wonderful Period 3 AP Bio class. Say hi.
Hi.
All right, and we are going to pick up at the reproduction of a virus, but before I do that, could I very quickly, oh, you can't see what I can see. I want you to go over the structure of a virus. What are the components, and why don't we have the oldest bio buddy do that? Go ahead, oldest bio buddy. Talk about the structure of a virus.
Has some kind of nucleic acid. It can be produced by human
I know you did this one. You should get 100%. No. I just need to get your brain in the right spot.
Oh yeah.
Is that it? 1738 number? You remember that because it's naughty.
Just a number in a song. It's like a time.
Who sings it?
I don't
Very well.
Who?
It won't let me sign it.
I don't know who that is. I know you're surprised.
I don't know either.
Look at this.
Waiting for one more. YG. Oh, hi, Juicy J. 5, 4. Commit.
It's a weird big. He's blind. Not that eye.
Oh, very disappointing. Good job, Jay-Z. Let's see who we have trouble with. ASAP Rocky with $. Okay. So they forgot D. Yeah. So classified whether or not it has that outer envelope. Okay. We are moving on to their life cycle. I'm going to start with a bacteriophage. How do you know I have a picture of a bacteriophage here? Does it have a recognizable structure? Who are some people who did an experiment with bacteriophage? Good. And they knew that there were 2 parts to the nucleic acid. There's always, dang it, 2 parts to a virus. What are they? Nucleic acid and a protein capsid. In the case of this bacteriophage, it has to attach onto a bacterium, then it injects its nucleic acid, and so that part is called attach, and then penetration is when it injects it. And then biosynthesis, what happens is it takes over the host cell's machinery to transcribe and translate its nucleic acid into more viral components. Maturation, you're putting all those viral components together, and then release, in this case it's lysing the bacteria, killing the bacteria, and thousands of viruses are released afterwards. The way I like to remember this, so you remember the stages, is APBio Ch. You see it? Okay. Let's go over, make sure we have some good notes on that. Attachment, it's a host-specific match. Host-specific match. Penetration, now there's different ways. In this case, this bacteriophage, it is injected, but it could be engulfed or injected. Engulfed or injected. Biosynthesis is hijack host machinery to synthesize viral components. Hijack host machinery to synthesize viral components. Maturation, assemble new viruses. Assemble new viruses. And the release is exit through lysis. In this case, it's lysing it, or budding. Or budding. Okay, and then if you can see on your notes, it says variations on viral reproduction. This is the basics, okay? For bacteriophages, and there's other examples as well, but I'm just focusing on bacteriophages right now. There are 2 alternative life cycles, the lytic and the lysogenic cycle. And in its simplest explanation, the lytic cycle is exactly what you saw. Yeah, attachment, penetration, biosynthesis, maturation, and release. This is the lytic cycle. But sometimes following attachment and penetration, it does integration, where its chromosome is integrated into the host cell chromosome. Remember, if it's a bacteria, it's a single circular chromosome, right? So it integrates into the host chromosome, and then it's called a prophage. I like to think of it as a professional virus, a prophage. And it integrates every single time then that host cell replicates. All of its daughter cells will have a copy of the virus integrated into the host cell chromosome. And then at some point, it can re-enter the lytic cycle at biosynthesis. This type, where it lays in wait, that's called the lysogenic cycle. In lysogenic, what do you see? What's this word? Genic, what do you think of? Genes, or genetic. So it's in the genes at that point as a prophage. Right? This is it, a little more detail. I want to have youngest bio buddy take the lead on this, and then we'll take a few notes on it. Go ahead.
But what happens after you first have to attach it to the DNA. And once it's attached to the DNA,
this is where it goes here or here. It goes, it's either going to go here or here at that point, one or the other. It's not one comes before the other. It's like either going to go here or here. So once it's here, it's going to stay there until it jumps back in at biosynthesis. All right, so let's take some notes on that. The lytic cycle, standard 5 steps. Host cell is lysed at the release by virally encoded enzyme called, ready? Lysozyme. Lysozyme, L-Y-S-O, zyme, Z-Y-M-E. What does that sound like? Lysosome. What do we know a lysosome acts like in a eukaryotic cell? Stomach, breaking things down. Okay, a lysozyme. The lysogenic cycle can happen following penetration. You will have integration step, where the viral DNA is incorporated into the bacterial DNA. Virus is latent and is called a prophage, which is replicated along with the host DNA. Replicated along with the host DNA. Environmental factors like UV radiation can trigger prophage to re-enter the lytic cycle. Sometimes the prophage causes the bacterial cell to produce a toxin, which can be bad for us. So which one is quicker? Lytic, yeah. So it comes in, replicates, busts out. Lysogenic, it's laying in wait. More insidious, right? More trying to hide. We know a virus like that. What is that? HIV, exactly. Okay, so now we're going to start to look at some other modifications. So here we looked at a bacteriophage, the lytic and lysogenic cycle. So now let's look at an animal virus. And oldest bio buddy, I want you to take this one and talk about how is it similar to what we've learned? How is it dissimilar? How is it different from the bacteriophage? Go ahead, oldest bio buddy. How is it similar? How is it different? Top left down to the bottom right. Go. So tell me wise ones. How is it similar? The what? The DNA what? The viral DNA is replicated. It's transcribed and translated as well, right? you're still making more viruses, right? You're still taking over the host cell machinery and getting them to produce more viruses. How is it different? Yeah.
The entire virus goes in through endosome.
Entire virus goes in. How else is it different?
It has a whole assembly process and budding.
Okay, so the assembly process is going to be similar, right? But budding, this is important. What's happening when it's budding out of the cell? Instead of, we saw lysine before. It killed the cell and came out and destroyed the cell. Okay? In this case, it's budding out. What is it acquiring when it's budding? Yes? The animal cell's membrane. Ah, why would that be important? Yeah, it can what? Hide. Pay no attention to me. I'm just a tiny little cell. Okay? And can I come in? Because I know the secret knock. See, I'm one of you. I have similar proteins. Now, it will also have the proteins, the like viral spikes it can have. In fact, it gets those synthesized as part of one of the things that gets translated. It will produce these viral spikes that allow it to gain entry. Gives it the secret knock to get into our own cells. And we go, oh yeah, come on in. Okay? Now, antiviral drugs, when you're taking those for whatever it is, a lot of times they could interfere right here with the biosynthesis part. So you're not getting rid of the virus, you're just not allowing it to make what? More of itself. And that's why some of them are very time-based. You have to take it at the same time every day, because if you skip, then it can go on to the what? The next stage that it's going through in the viral life cycle. So you never want to skip. You want to always take it continuously. All right? So for our animal virus, little letter A is talking about all the different ways it can gain entry. But either way, it has to be uncoated because you have to remove the cell membrane, or the membrane that it stole from the last cell. You've got to take that off. You've got to take off the protein capsid. And then you can enter into the lytic or lysogenic cycles. Enter into the lytic or lysogenic cycles. And then B, it buds from the host using various membranes. It could use the cell membrane, the Golgi membrane. It could use a nuclear membrane, or just lyse host if naked virus, or just lyse host if naked virus.
What's A again? Sorry.
Lytic or lysogenic cycles. Who just explained this one? Young or old? Oldest? Okay. Young, we're going to look at another modification of a viral life cycle. Young one, you explain about a retrovirus. What's going on here?
First it enters the cell, and
Okay, now I'm going to back up for one moment. Okay, when we looked at this, okay, this right here could be DNA, or this right here could be DNA, or it could be RNA. Okay, that you're using, that some sort of nucleic acid message to make more of that virus. It could be single, it could be double-stranded, all types of options. In this case, though, we have an RNA virus. And this RNA virus, okay, once it still gains entry, we're doing, you know, you're getting brought into the cell, you still have to take off the cell membrane, you still have to take off the capsid. But then it is an RNA virus and it's single-stranded, and you're going to use what enzyme? You already know, reverse transcriptase, to make a single-stranded RNA strand. And then you can use like replicase. It's another enzyme to make it, instead of being single-stranded, it would be what? Double-stranded. And then you need to use integrase to sit and get inside the host chromosome. Now, if you went out and got HIV today, you went down to Hollywood, slept with 25 people without any protection, that'd probably increase your risk of that, yes? And you'd be really tired. But if you did do that, you would end up getting sick, like the flu, if you got HIV, just like you would any other kind of cold or flu virus. And your body would take care of all the free-floating HIV viruses that are in your bloodstream. It would take care of cells that are showing signs of having HIV through cell surface markers. And you would wipe it out like you would any other kind of flu or cold, and which we'll talk about more when we talk about the immune system. However, it can't wipe out your cellular defenses, cannot wipe out something that is what? Hidden. Okay? Because if it's out and in the cell, you can't help it. You just like keep displaying what's inside. You're like, this is inside of me, kill me. This is inside of me, kill me. But if there's no virus aftermath to display, your body doesn't know that it's hiding within your own chromosomes, and so it doesn't kill it. And every offspring cell, every daughter cell will have that same HIV virus in there. And that's why you can go, and it acts as a prophage, you know, and it can live for years and years, 10, 15 years. Then you have some major need of your immune system, something triggers your immune system, and then it's like probably some sort of adaptation of the virus, like, ooh, bad things are happening here, they're sick, we better find a new host. So it moves out of its prophage, moves out of that lysogenic, maybe more into that lytic cycle like a bacteriophage. It comes out, it starts to do biosynthesis, making more viruses, and then you release them. Okay? All right, so for retrovirus, animal virus like HIV with RNA genome that is converted into DNA, what enzyme are you going to put there in the highlighted area? Reverse transcriptase. It can remain latent for years. Antiviral drugs interfere with steps of the life cycle. All right. Now, there are many human viral diseases that you can get. Pick your top 3 and share them with your bio buddy. Go ahead. Now, all of you have been vaccinated against chickenpox, right? Yeah. So that kind of just started happening. Well, not for you. Let's see. That'd be my oldest son is. My oldest son is 22, and it just started happening around that time that you got to have it. We did not get to get vaccinated against chickenpox. If one of our friends got chickenpox and it was like summertime where you wouldn't miss school, then our parents were more likely to say, "Ooh, let's go play with Nancy today." And then they would want you to get infected, so then you would get chickenpox over with, because once you got chickenpox, then you were unlikely to get it again. But I know somebody who got it 5 different times. And the reason is your immune system can become hyper-specific to a particular strain of it, and it wouldn't recognize the other strains. Every time it thought, oh, new disease. And then, so it couldn't fight the infection, and then they came down with chickenpox again, so 5 different times. But a lot of times, it's just once and done. Some of your parents might even have scars from chickenpox. If you ask them if you have any chickenpox scars, I think I have one on the front of my shin from chickenpox. But that's just not something you experience anymore. Bless you. The concern for your generation and my generation are these emerging diseases. And the reason why is some of these are connected to climate change, why we still believe in climate change, at least for the next 10 days. So these emerging diseases are appearing for the first time. Either they were in a limited area that we weren't exposed to them, And we're doing things like expanding into certain forested areas where nobody lived there before, and they're cutting them down, and so they're getting exposed to them. Some viruses can species jump now, so they can go, like where I told you would never get tobacco mosaic virus. It's not like I came down with the tobaccos. I've started smoking, you know. It's not like that, but some have acquired the ability to do species jump. Through climate change, you create different conditions in different areas where you wouldn't normally have to be concerned about a particular virus or pathogen, which now we do. On your notes, for emerging viruses causing new or previously uncommon illnesses, for it to be called an emerging disease, they usually say it's a widespread disease, widespread, like HIV, West Nile virus, hantavirus, Ebola. You know, a little liquefaction of your organs, no big deal. The virus is extending its range, it's gaining access to new area through transport. So as our transport systems have increased, you know, somebody can jump on an airplane and take that virus from one continent to another just like this. Or genetic mutation, increasing their virulence factors. Some mutations allow viruses to species jump. Species jump. So there's a couple words, pathogenicity and virulence. So pathogenicity is, can you cause a disease? And then virulence is a measurement of what it does, the degree of pathogenicity, how invasive it is, how toxic it is, how quick the death is, its ability to change or mutate. So on virulence, quantitative measure of pathogenicity, and pathogenicity is the ability of an organism to cause a disease. If there's a new disease but it's fast acting, if it causes you to die within maybe 48 or 72 hours after exposure, that can be a good thing. Bad for the person who has it, but it's easy to be safe because you can quarantine that area. Whichever ones die, but then it doesn't spread, right? You'll know if it's spreading. It's some of the slow-acting ones, the ones that become prophages that can stay within a system and spread without people's knowledge. Remember, like what I told you about, like the cold sores I get on my lip, right? That gets a lifetime. Now, obviously, it's virulence. It doesn't kill me to have a cold sore. It's just uncomfortable, right? But if there was something like that that was slow-acting like that killed you, that would be really bad. So the concerns, viruses can evolve so rapidly, that's why you always have to get a yearly flu shot, because it's a new strain of the virus. It makes it difficult to find cures. And you have a highly suggested reading and thinking on that one, on the evolution of the flu viruses that you're going to look at. Okay, and then infectious diseases are sensitive to climate, and this is from the Harvard School of Public Health. Are sensitive to climate, so as the climate changes, so too will the diseases. And then I give you a link to research at Harvard. Okay, so in this case, the reason why this spreads is as the temperature changes, things with malaria and dengue fever. Dengue fever is like hideous. I've never had it, I don't know. But dengue fever, the mosquitoes will be in areas like tires that have sitting water in it. Mosquitoes then can use that as a nesting site. And you have, if I remember correctly, I mean, you could Google the symptoms, but I think you have incredible bone pain and diarrhea, and you want to die. And I've heard people who've had dengue fever say, I'd rather die than get dengue fever again. And other people have more milder forms of that. But it's transmitted by mosquitoes, which can't survive if temperatures are too low. So if you have climate change and you have areas where the temperature is elevated, then you have groups of mosquitoes that can live there, and if they're carriers of some of these viruses, that can be very bad. Okay, there are other items too that cause disease and pathogenicity, and that would be, one is a viroid. A A viroid is like a little naked strand of RNA. Do you remember we talked about small interfering RNAs? Remember that? Okay, and so we talked about it in transcription and translation. We talked about that process. Viroids, these little pieces of RNA, can interfere with crops. So this is the one on the right has a viroid and the one on the left doesn't. And they're kind of like bad friends. They influence others to go bad as well. And so one bad RNA can get the cell to make more bad viroids, and it can just decimate a crop. So naked strands of RNA, it directs the cell to produce more. So the viroids are different from a virus. Why? How are viroids different from a virus? No capsid, exactly. These are just straight up little RNA fragments. Okay, and now these are prions, and these are proteinaceous infectious particles. So what do you hear in proteinaceous? Proteins. These are rogue proteins that cause diseases. Once infected, it starts a chain reaction that converts other normal proteins to prions. Starts a chain reaction that converts other normal proteins to prions. It's the cause of TSE, transmissible spongiform encephalopathies. It causes little holes in your brain, makes it spongy in appearance, and this would be similar in cows. It's called mad cow disease, but in us it's TSE, Creutzfeldt-Jakob syndrome. Okay, how are these different from viruses? They're only proteins. Okay, and then viroids are only RNA, and a virus is a nucleic acid with a what? Protein capsid. All infectious, all bad. All right, yay. I want to hear Young Ming Ming rap. Somebody needs to pull that up so I can hear it. Young Ming Ming. Let's go, Jay-Z. Oh, good job, Notorious B.I.G., Dr. Dre and Drake. Okay, so yeah, why did you miss that? Some of you picked a lytic virus as a prophage. No. Which word is shorter, lytic or lysogenic? That's the quick one. Okay. Lysogenic, we hear in that word what? Genes or genetics. It's in the genes. Okay, that's a prophage when it does that. D doesn't have anything to do with being an envelope virus. A prophage is not an envelope virus. Check your BioBuddies clicker. Give them a gong of victory or a little bell of shame. Ring that shame bell if they missed it. In case you don't know, these are the ones that need the shame. Jay-Z, Nicki Minaj, for many reasons. Big Sean, G-Eazy, Rocky, Ghostface Killer.
No. I need you.
Relax.
Do you see a good essay question in what we just went over?
Oh, I spelled it wrong.
Anybody?
It's like on the 3-5 game, the old leg is gone. When I need it, oh gosh.
Hey, anybody see a good essay question in what we just went over? What? Or comparing and contrasting the lytic and lysogenic, or comparing and contrasting a bacteriophage with an animal virus, with a retrovirus. Those are all great questions. Ghostface Killer. Let's go. I have think we had some spelling issues, but we were trying to get there. We were trying to say biosynthesis.
What was it?
Oh, I'm sorry, yes, let's see who's first, by all means. Dr. Dre. Was that you? Yeah, Dr. Dre, of course you are. Dr. Dre has really made, the one that has made the most money out of everybody from NWA.
I think it's a B.
And producers.
Yeah, he's a big producer, huge, very gifted. Not that I'm into that, but
No, he is though.
yeah, he really is.
And he donates a ton to USC. He's donated like $5 million.
Oh, okay. That's why I think it's C.
Who's already answered the question? Look what I made. I'm all excited. I thought of this activity over winter break. It's titled, Honors is gonna do it, but I have enough licorice, I can have you guys do it too, as a review. If chromosomes were licorice. See, and the two on it, see, and you can make sister chromatids, see it? Do you like it?
I love it.
And you could like do it and then eat it.
Wait, so this.
Oh, losers, please. Polymerizes host DNA. What does that mean? Let's just think what those words mean. Polymerizes means to make more of the host DNA. Do any viruses do that? No, shame. And then 5 of you picked D, translates host DNA. Do viruses translate the host? No, they have the host cellular machinery take over their nucleic acid. Look at your BioBuddies clicker. If they don't show, they don't know, and just start ringing that shame bell. Otherwise, give them a gong. Okay, and I'm going to stop this video here, and then we'll do a re-recording. Hope you're having a good day. Have some toast. Just wash your hands first.